Good afternoon, everyone. I am your host, Shailesh Shukla. We will be starting this webinar very shortly. Thank you for your patience. We'll be starting the webinar very shortly. Thank you for your patience. So again, good afternoon, everyone. I hope you are doing well. On behalf of Advent Informatics, I, Shailesh Shukla, warmly welcome you to today's webinar on a focused and highly valuable topic, which is post docking analysis, and graphical data retrieval using Molsoft tools. So we are delighted to have you with us and looking forward to a session full of practical insights and interactive learning. Before we get into today's session, allow me to briefly introduce the organization behind this event is Advent Informatics Pvt Limited, headquartered in Pune. Advent Informatics is a leading distributor of advanced life science software solutions. Our core strength lies in chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and biological information management. We cater a broad spectrum of clients, from top pharmaceuticals and chemical companies to CROs, which is contract research organizations, and academic institutes, plus the government R&D labs across India. Our mission is to bridge the gap between scientific challenges and software solution by making world-class technology easily accessible to Indian researchers and professionals. We are proudly representing several globally recognized technologies partners, including you can say Molsoft and Chemexon, Campus, Satera, Transin IT Solution, and Key Module. With their support, we continue to drive, discover, and innovation across life science domain. So now let me introduce our speaker. So please welcome Mr. Aditya Mishra, a junior application scientist at Advent Informatics. Mr. Mishra has built a strong reputation in the field of computational chemistry and molecular modeling, with a focus on practical applications enduring drug discovery and design. At Advent Informatics, he specialized in enabling scientists to efficiently use platforms like Molsoft ICM for tasks including virtual screening, docking workflows, various bioinformatic and chemoinformatic tools. His deep knowledge of drug designing domains and hands-on experience with the tools like ICM make him the ideal presenter for today's session. So please join me in welcoming Mr. Aditya Mishra as he's guiding us through the essential of post-docking analysis and the graphical retrieval using Molsoft tools. So before during webinar, if you have any questions or queries, so please feel free to type them in the chat box. So yes, Mr. Mishra, the floor is yours.
Thank you so much, Shailesh, for the wonderful introduction, and very good afternoon everyone present here. And like Shailesh introduced me, my name is Aditya Mishra. I'm working as a junior application scientist with Advent Informatics. And today for the webinar, we have taken the topic, which is post-doc analysis and graphical data retrieval using Molsoft tool. It is in continuation with regards to our previous webinars. Firstly, there was a webinar on molecular docking and virtual ligand screening, and now we are continuing that with post-doc analysis and graphical data retrieval tools. Before beginning the webinar itself, let me just introduce the company and where I come from. So we at Advent Informatics, Advent Informatics was founded back in 2016, and we are the prominent distributor of molecular modeling, bioinformatics, cheminformatics solution nationwide. We are also the first level of technical support what all our clients are getting, and we are highly appreciated for the same. Last year, we conducted a survey about our responsiveness, and we got 5 out of 5 star for that. And there was also a question how prominent we are while responding to the queries raised by our clients. 50% of our clients agreed that we respond to their queries within 1 hour, and rest all 50% agreed that we do it within 24 hours. So we at Advent represent different software solutions. Firstly, we have Chemexon. Chemexon is a global leader in cheminformatics, chemical data, structure and enumeration, library enumeration, prediction, and all such things. We also represent Campus. Campus is a AI-assisted lead discovery and forward reaction prediction tool. If anyway you are into synthetic chemistry, you can definitely look in detail about Campus. We also represent Key Module. Key Module is a tool which is able to extract chemical structure from any document, whether patent, publication, and any such thing, and not just extract them in editable format. We represent D360 solution from Satera. It is basically a data visualization and analytical tool. We have Transin IT Solutions as our partner. It is a Bangalore-based company, and it is into ELN, compound data management, and all these things. The latest addition in our partners is SCM. SCM is a tool which is mostly into material sciences, computational chemistry, quantum science, and fluid thermodynamics, all these things. We also represent Molsoft. It is a complete molecular modeling suite, and we are going to see in detail about this tool today. Going about the journey, what Molsoft has even till now, it's like Molsoft was founded back in 1994 by Professor Rubina Pigyan when he came up with two new technologies. First is ICM, internal coordinate mechanics, and bias probability Monte Carlo methods. Right. So on basis of these two technologies, the foundation of this company was there. And in the decade from 1994 to 2000, there was some major collaboration with big companies like BMS and Novartis. And this collaboration yielded tools like with partnership in SGC, we got Scrap and Active ICM. And with partnership with Novartis, we got tools like ICM 3D ligand editor. So all these points, what we see in purple here, these are some of the success stories of what I'm going to briefly explain you during the webinar. And also in the same decade, we introduced a new technology, which is atomic property field. It is kind of a pharmacophoric grade potential tool. So talking about success stories, in the decade of 2000 to 2009, we participated in a GPCR challenge where the challenge was to predict the structure as well as reproduce the crystal pores. We were the top ranked commercial tool in that. We were also the first ranked tool in Capri challenge. In the next decade, from 2010 to 2009, there was some major development, and deep learning based tools like Molescreen was launched. GigaScreen was also launched in collaboration with Inamine databases. We got a new FDA approved drug Pepomir by Rempex Pharmaceuticals, and the designing in silico part was done using ICM tools. There is a D3R docking challenge where both commercial as well as in-house built tools can take part. And for 3 consecutive years, ICM was top ranker in that D3R challenge. So where in this decade, it was a new era for cloud computing. So what Novartis did, they undertook 30,000 cores from Amazon clouds, and they did a 39-year worth of computation within 30 hours. And as a result, they got 3 lead compounds. Also, there was a docking, covalent docking challenge, and ICM was the top ranking tool among them. In the current decade, from 2020 to the current year 2025, where it is the era of AI and ML, Molsoft is not behind, and they have introduced new tools like RIDES, Rapid Ancestor Discovery Engine. It is able to like screen 6,000 conformers per second in a normal CPU, and if we talk about GPU, we are able to screen 1.5 million conformers per second using a normal GPU, right? We have introduced a new scoring function, which is RTCNN. It is a neural network based course, which we use during our docking and all such things. There are major platforms which are using ICM tools as their primary discovery tool. For example, Arkansas Therapeutics is there, Alien Therapeutics, Launch platform, and all such things. Also, in 2021, there was a study where 11 billion chemicals were screened, and not just screened, the screening was done in Synthon-based approach, and the ICM tools which were used in that, and that specific publication had impact factor of around 69 point something. Talking about AI ML tools, we do have some add-ons. Firstly, we have Rich. Rich is a rapid docking GPU engine. It is able to dock 100 compounds per second in a GPU, which is RTX 4090. It is commonly found in a gaming GPU. It's not a very high-end GPU. Right? It is the fastest structure-based screening tool as per our knowledge, and it is able to screen up to 10 million compounds in less than 30 hours. We also have GigaScreen. GigaScreen is used to screen to giga-sized libraries like billions and multi-billion compounds. It uses Rich as its primary docking engine, and it works on active learning. We also have Ginger. Ginger is a high-quality, lightning-fast conformer generator tool. So talking about global users, and this is just a subset of that. When we talk about Molsoft users globally, we have big pharmas like Pfizer, Novartis, Eli Lilly, Vertex, Merck, AstraZeneca, Arkansas Therapeutics in terms of commercial users. And if we talk about academic side, big names like New York University, Stanford University, NCI, University of Southern California, all these are using Molsoft tools. when we talk about Indian users in commercial side, we have Syngene, Origin Oncology, Origin Pharmaceuticals, Zydus, Think Molecular, Eurofins. These are on commercial sites, and in academic side, we have names like NIPA Guwahati, Geetam University, Chanakya University, Central Institute of Kerala, Bharati Vidyapit, VIT, DYP, and list is we have long list. We are only representing Molsoft in academia in India from last 2 years, and we do have a good client base on that. Talking about modules and functionalities, what Molsoft offers. So firstly, the flagship product is ICM Pro. It is the base model which is able to do pretty much about structure-based modeling and all such things. With ICM Pro, you can have add-ons which are available. Firstly, we have VLS module, we have homology module, we have ICM Chemist and ICM Chemist Pro module as well. So VLS module, if someone is using, they have all the features in ICM Chemist and ICM Chemist Pro add-ons as well. We do have some web-based solutions as well, ICM Browser Pro, Active ICM. So most of them are free to use when you talk about web-based solutions. So typically, when we talk about the combination, which is most effective, if a user has ICM Pro along with VLS and homology add-on, they are able to do most of the screening part in terms of the in silico journey, from hit to lead identification to all the way to clinical trials. All these points which I've highlighted in green here, you are able to do it with the current combination of ICM Molsoft tools. Okay. Moving forward for today's agenda, where we are going to do post-docking analysis, what are the things that we are going into, right? Firstly, we'll understand what post-docking analysis aims to answer. What is the meaning of it? And then continuing with residual level determination of binding, what kind of binding, non-binding interaction are there, their significance, all those things. We'll also see interaction profiling and visualizations option, what we have. We will also do a case study during about our demo, and we will desert our good fit or bad fit around our VLS. We will also understand what are false positive, how to identify and screen them. And at last, we will understand the practical workflow, which is best for the practice. So the first things first, we need to understand the process of docking and why we are doing to post docking. So it is a journey. Firstly, you will do docking, where you'll do the simulation, predict the poses, rank them on the basis of score. This is the docking part. After docking or post docking, we need to validate or refine our results. For that, we need to understand few things. Are key pharmacophoric interactions present? There are residues present. They need to make those interactions. We need to have a certain groups. They should be chemically and strictly plausible. And also, do the top hits in our scoring, do they agree, or do they perform well when the protein is more dynamic, right? So giving a term good fit, it means there are multiple complementarity which needs to be completed. Firstly, we need to understand the shape complementarity. The shape complementarity basically means the volume what the receptor pocket has and the ligand which is fitting inside it. Secondly, the chemical complementarity is when the donor atoms or donor part of your receptor meets to your acceptor part of your ligand small molecule and vice versa. We need to look into that. Also, we need to balance the charge of both sides if you have charged moieties inside your object. We need to understand hydrophobic matching. So all the apolar regions in your small molecule should buried in your buried in apolar regions in your receptor. Right. We need to understand the importance of water network, because water of high energy needs to be displaced while the simulation, while binding, and we also need to respect the waters which are inside binding pocket, which might be necessary for like say water bridging and all such things. Also, we need to consider a protein as a dynamic entity, so it should we should consider the side chain flexibility, rotamers, and all such things. Okay, so all of this we are going to do with ICM tools, and before that, how we are going to do it, and we need to understand the technology behind the tool, right? So ICM stands for internal coordinate mechanics, and typically where other software are based on Cartesian coordinate to determine the position or change in position of any entity, we do that with different variables. right? These are internal coordinates, and these consist of bond length, bond angles, torsion angles, and phase dihedral angles. In normal conditions, all other variables are constant. The only change is in torsion angle, and this technique can be utilized using in protein folding, protein modeling, docking, virtual screening, and all these things. The second technology what we need to understand is biprobability Monte Carlo methods. So this is a sampling algorithm in which there are 4 different steps. Firstly, if you have a structure, then random conformational changes will be made of that structure. Next, the local minimization will be done of that structure. In third step, additional energy terms will be calculated, like dissolution energy, entropies, and all such things. And in the last step, we will calculate the total energy of that specific system and rank our poses on that basis. And you can utilize either the top one of the conformer or a set of that, either 3, either 5, whatever. So in our previous webinar, we got to know about small molecule docking, how we can do it using ICM, and also we did a VLS. So this was the normal workflow what we did. Firstly, we prepared a protein in our ICM that is basically called converting it to ICM object. Next is we generated a grid, we ran the docking simulation, we analyzed the results on the basis of scores. And for today's webinar, we are going to highlight these 2 points, analysis and validation part. So firstly, understanding there is a binding pocket and your small molecule is binding inside it. So there are bonding and non-bonding interaction. Firstly, hydrogen bonds are a necessary, let's say, interaction where we need to understand the donor, acceptor. And if we are talking about hydrogen bonds, we need to know the neutral and charge-assisted hydrogen bond. They both exist. Their geometry in terms of angle and distance, they do matter while considering the hydrogen bonds. Also, if there are salt bridges, you should know the oppositely charged pairs should be paired together. Aliphatic or aromatic side chains of your small molecule should be buried very well inside the pocket, and it should we should understand the buried solvent accessible area, that SA here. So these are the points where we say the more buried surface solvent accessible area is there, the more binding affinity is towards that small molecule. Okay, metal coordination, the metal coordination states, the charges, the geometry, all of these matters when we are considering a pose. Right. So this is a table where where I have just represented the energy ranges of non-covalent interaction, because if you have covalent interaction, those are completely different. These are important, and mostly they are irreversible interactions. So these are relevant to hydrophobic and electrostatic hydrogen bond, pi-pi stacking, van der Waals interactions, and these are the ranges. Talking about practical geometry of the bonding interactions or non-bonding interactions, firstly, talking about hydrogen bonds, let us understand how we represent them. In ICM, we represent hydrogen bonds in spheres, right? So the size and color of those spheres generally shows, tell us about the strength of that bond. Typically, when the hydrogen bond just the length ranges less than 3.2 Armstrong and angle is more than 140 degree, we call it a good interaction, right? Salt bridges, heavy atom distance should be less than 4 Armstrong. Aromatic stacking, the centroid to centroid distance should be around 4.5 to 5.5 Armstrong, and offset of 1.1 to 2 Armstrong is acceptable. If we're talking about cation pi interactions, so the ring centroid should be around 4 to 6 Armstrong. Steric heavy atom clashes. Right. So talking about steric heavy atom clashes, there is a possibility then your heavy atoms are getting clashed during the movement. Right. So we need to check them. We also need to consider the bumps in the map. Torsions, we need to consider torsions as well to avoid eclipsed or strained torsions, and also we need to compare to Rotamer's library if there are any. Sampling double bond in our system is necessary to consider resonance in that molecule or set of molecules. Talking about the surface, the binding interaction, the residues around it, hydrogen bonds and all such things, we need to understand we are considering the pocket to be important moiety which is interacting with a set of small molecules there, and each molecule may need to be optimized. And for that, we have a very good tool with ICM, which is ICM Interactive 3D Ligand Editor. With the use of ICM 3D Ligand Editor, you can optimize your small molecule inside your binding pocket. You can see the clashes if there are any. You can make the changes in 3D as well as in 2D. You can also use the same tool for multiple purpose. For example, use the best fungi replacement group, do multiple position R group scan, do bioassisted scan, also scaffold hopping, core replacement could be done. Here I have a little video where I can show you how the tool is trying to fit in a functional group and making it to minimum energy state. Right. Going forward, understanding the residue, the geometry, their type is very necessary, right? So there are different type of residues when we are talking about a pocket. Firstly, we have anchor residues, which are the conserved residues, and those are different the residues which define the pose. We have selectivity residues that are that basically variable position shaping subpockets they can give due to the selective residues, right? Next, what are gatekeeper residues? Gatekeeper residues are generally those residues which are in the entry site of your binding pocket, right? So by entering, being there, they can control the access of your small molecule to enter into the pocket. Also, by their movement, they are dynamic also. So they can change or let's say modulate the volume of your pocket. And often they are the reason for the clashes. We also need to understand the water mediate contacts, the water bridges networks for the stability and replacing the high energy waters and all these things. Another good point is, like I was saying previously as well, that dynamic protein is a dynamic entity. So we need to consider side chains to be flexible. Those side chains will either adapt to the ligands, or they might be in place, give you some clashes and all such things. So next, talking about different post-docking analysis, tool, not a tool, a factor which means a lot here is enrichment factor. Enrichment factor is basically the presence of biologically active molecule in your top scoring candidates. It is basically calculated on the basis of, for example, I have 1000 compounds in my library, and I know 50 or 60 of them are biologically active, and I do know they are active and their activity values as well. I ran a docking simulation of the same library, and top X%, this X would be 1%, 2%, 5%, 10%, whatever it is. In that X% of my top scoring candidate, how many active moieties are there? The percentage of that biologically active compounds in my top candidate, top X%, is generally termed as enrichment factor. Talking about enrichment factor, there was a publication from AstraZeneca where they have compared different tools for post-prediction and enrichment factors, where they have compared ICM, Glide, Gold, Plexus, and visually it is clear that ICM gave the best RMSD among all the available tools. And also it was mentioned that on target by target basis, Glide would appear to be a choice of tool in 4 family classes, whereas ICM would be a better choice for 8 different family classes. In the same study, it was highlighted that ICM has the best enrichment factors among all the available tools. Okay, false positives. The term itself defines a lot, but to give a brief about it, it is a common observation when we run a docking simulation, we might not get biologically active molecule in our wet lab assay when we look for top scoring docking candidates, right? So this could be a result of being false positive in your docking results, right? And where they come from, we need to understand. So one of the reasons of false positive is scoring function. They are biased. They are too simple for that. Scoring function are based upon algorithms, and they are not yet fully capable to understand the biological system and give the right result, which are 100% sure. Right? The next point is to consider protein, not to consider the protein to be dynamic. If you're using a rigid protein, you're neglecting multiple things. For that, if you want to eliminate those things, what you can do, you can look for multiple receptor confirmation. Those can be either cryo-EM models or snapshots from your MD simulation, or you can look for induced fit docking or the side chain sampling. All these techniques you can use here. We can also keep the hit consistent across relevant state of the protein. Another reason is ignoring misassigned protonation, tautomers, or metal state. considering charges and everything are important when looking for good pose. Overfitting to artifactual subpockets. So artificial subpocket are those pockets which do not exist, but they are in a result of crystallographic noises during the crystallography process. Okay. And also we need to consider poses which are only viable in rare or unrealistic conformers. They do not really define geometry and do not exist practically or science-wise. They are not very much possible. So we need to take that in account as well. Previously talking about induced fit docking, with ICM you have three different approaches to deal with induced fit conditions. Firstly, we have 4D docking, where we make an ensemble out of multiple conformations of the same protein, and then after we make that ensemble a receptor, we dock the library or small molecule in that ensemble, multiple receptor ensemble one, and at last, if the docking simulation when we are doing, when it completes, the algorithm will tell you a specific pose is getting bind to which specific conformer of your protein, right? Second, we have hybrid partiality explicit method, by which we allow some of the residues to be flexible. And the third approach is to look for scare method. So scare method, like I explained about the gatekeeper residues in previous slide. So this is about somewhat about that, where we replace the heavy amino acids on the pocket entry sites with lighter amino acids, like in this case alanine. We place our small molecule inside that, it makes the geometry and it will fit inside it, and at last we'll replace the original amino acids and see the interactions there. So how we can screen false positive? Firstly, always physicochemical considerations are there. You need to consider all the physicochemical sanity here. Log P, PSA, hydrogen bond donor acceptor, basically Lipinski rule and all such things. 2D alerts from cheminformatics side, like paints, reactive oxidative species, aggregators, filters. If you have covalent warheads in your system, you need to consider them. If you don't, it will not make the good fit. Conformer or strain. The strain in your molecule depends upon how they are present in the 3D space, right? So energy strain less than 6 to 8 kilocalories per mole are relatively in a global minimum state. So post plausibility, like I said, which are possible and they are not showing any severe clashes and they are realistic, we should consider those poses. So with ICM, previously I was talking about scoring error. We have not just one, but three different kind of scoring algorithms. Firstly, we have ICM score. ICM score is a physics-based course where we consider different parameters like hydrogen bond donor, acceptor, solvation, dissolution, energy, size, and everything. Some of the parameters are somewhat similar to MMGB assay here. We also have MF score here. With MF score, it is a statistical knowledge-based score, and at last, we have the latest edition, which is a RTCNN. RTCNN is a neural network based score, and it was trained against native ligand and decoy complexes. So there was a study, internal study, where we compared RTCNN and all the available scoring function, and on that, while ranking top three poses or candidates in a sample set, RTCNN was the top ranking scoring function. It outperformed even our own physics-based scores, outperformed Glide score, Autodoc Vina, Glide XP, and everything, right? And in the same study, it was mentioned that RTCNN is giving us the best enrichment factor as well. So when we are doing all such things, the next step would be to go and look for MD-based validation. For MD-based validation, what we need to do, we need to run a small MD of your selected pose. And when we do so, we need to consider two, three things. Your ligand molecule or small molecule may shift out of your binding pocket. So considering RMSD is a good option there. You need to see the persistence of hydrogen bond throughout the simulation. This should be persist for more than 40 to 60% of the simulation time. Water behavior, like replacement of high energy waters and water bridges are considered in this part as well. If there are borderline cases, we need to consider longer MD simulations for 10 to 50. Reporting and documentation. So the second part of this webinar related to generating good quality images, videos, and such things, and taking a publication quality graphical output for the same. Talking about publication, if we just type about Molsoft in Google Scholar, we have more than 6,200 publication, and not just any journals. We have publications in reputed journals like Cell Death and Diseases, Nature Communications, Scientific Reports, PLOS Biology, International Journal of Biological Macromolecules, Blood, and all such things. Right. So let's just quickly jump on our demonstration part, where what we'll do, we will start with docking part and quickly do a VLS, or maybe use a file where I have already generated a result to do post-docking study. Right. For this study, I have imported a very interesting protein, which is 4R6E. It is a PRP1 protein, a very interesting target in cancers and such. It has a co-crystal inhibitor, which is nirapamid. And to begin with the docking process, first thing I need to understand that currently it is an X-ray crystallographic structure. It is in Cartesian coordinates. So what I'll do, I'll just convert it to ICM format, internal coordinate object. For that, I will convert it to ICM. And we have discussed all these options in our previous webinar. You can definitely go and check for the details here. Right. I'll just quickly make the changes, click on OK. Like again, this is the only protein preparation step you need to do while working with ICM. It took like 3 seconds and our preparation is done. Now, if I zoom in here, my co-crystal moiety is somewhere buried deep inside my pocket. And if I want to see the interactions, I can just go into the entry, which is A3DR here, go to pocket and look for ligand pocket interactions. As soon as I click into that, now I can see the hydrogen bond interactions in the system. The blue ones, green ones, the size, everything represent the strength of that specific hydrogen bond. Now, if I also want to see the pi stacking and all such things, I can just use this and I can see the stacking options here, right? Now, if I want to replicate the same pose, what I need to do, I'll just extract this from the structure here, and I can extract the structure in either 2D or 3D. Now I have the 2D structure here. In next step, what I need, I am going to make this binding site vacant. But before that, let's look for the 2D interaction, what this protein is making with the current ligand. For that, I can just go right click, go to tools, make 2D diagram. Auto detect is an option. You can search for, if you have multiple receptor in your system, you can search for them. Residue labeling option is there. Also, checking this table will give you additional benefit. You'll have a table with the residues and type of contact they are making, their distance, and all such things. Okay. So while we are describing about post-docking analysis, in between, I'll tell you some tools by which you can extract or take good publication quality images. Firstly, if I switch on this HD button here, I have a good resolution image. I can switch on this perspective button. I can, if I want to save the current 3D pose, I can just click into this button here, and it says copy image to clipboard. When I copy it, I can just paste it anywhere in my document, whether PPT, whether Word or anything, right? And I can do the same with my 2D diagram. Save or export, save image to clipboard, and you can paste it there as well. Okay. Now, for docking process, I just need to move it out of the pocket now to make the site vacant for docking. For that, I can just go right click, go to move from object. And now there are no interactions. I'll go to docking, make a new project, name it anything. Let's name it 4R6E. And you can browse in where you want to save that. There are different options regarding to identifying the binding sites. For now, I'm just going to use the simplest one where I have already a co-crystal ligand. Just selected it and clicked on define site around selected residues. 24 residues are now selected, and I can just keep this checked. Okay, and now I have a green surface life structure around my docking site, and In center, I have a yellow centroid. The centroid is basically the reference point where my docking simulation will begin. From here, I can click on go, and in next step, I can see the grid, right? So the grid, if you're satisfied with this size, that's fine. If not, you can just drag it from any of the corner and it will show get smaller or larger depending. Now, I have already done this docking part. I have also run a VLS out of it. And for the for today's demo, what I'll do, I'll just open that session where I have done docking and everything, and we will do post-docking analysis from there, right? So this is the same object where I have docked 500 compounds, and where I got them from, I can directly go to Molescreen here, go to find Campbell pKri data, type in the keyword that is PRP1, find. If I check and get Campbell pKri data, I had more than 5,000 molecules or compounds with it, with their activity data, activity value and all such things. Out of those, I've selected 500 and I ran a VLS out of that. Okay. So now, talking about post-docking screening, first thing what I will do is to rank my table or results in terms of score. The least scoring or the best scoring candidate, I'll keep on top. If I want to import that in my 3D graphical window, I can just single click here. Oh, before that, I need to import the same docking map. For that, you just need to select your protein or the maps where you have saved your docking. Okay, now when I click here, I have my 3D graphical window, and there I have the best scoring candidate, right? And it will be shown in M here in my workspace panel. Now you see that specific pose is making some similar interactions, and if I want to see them clearly, I can just go to settings here. I have tools panel. I can go to pretty view and visualize the current pose. I have options to reset or change the representation of my protein. Let's do it in ribbon form, color it by secondary structure, and also make it a little transparent for good visualizations. Okay. Transparent is the option here. And when I go to light, I can set transparency. Now, this is a better when I'm visualizing something. Okay. From here, I can see what the things I have mentioned about hydrogen bonding. I can see the distances. I can see what type of interaction they are making. If I have pi stacking in my system, I can take it from here. So let's do one thing. Let's compare different poses. For that, I'll just make a slide of current view. You can do it from here, add slide from current slide. And why we are doing it, I'll show you after we complete this analysis part, how we can use this specific file or slides in our Microsoft PowerPoint, right? So currently we have one view here, and you can save them with the same options, save to clipboard and all such things. And at the same time, we got to know about the surface, the complementarity of surface, receptor surface to ligand. If I want to compare the same and look visually into it, what I can do, I can just right click here, go to pocket, see the receptor pocket. When I do that, I have this pocket where ligand is very well buried inside my binding pocket. And at the same time, if I want to see the complementarity, so the colors of this surface shows us about the property of that specific region. The greens are hydrophobic regions, red are donors, the blue are acceptors. And if I make a ligand surface with the same option, you see I do have a mesh-like structure in same area now, and they are complementary to each other. Where we have the red regions in our receptor surface, we have the blue region in our ligand surface, right? And the green regions are common for both. So apolar regions are buried very well. So this is one way to look for interactions. Another good way is to just go here, and if you see, we have under tool, we have a panel here where we can calculate descriptors. We have property or expression calculation option here, right? So there are various options by which we can do post-docking analysis, and we can screen our docked poses, right? Firstly, we have charge. So charge does not only calculate the net molecular charge, it has the capability to influence solubility, influence membrane permeability, and electrostatic interaction with the protein, right? Let's just name it to charge and click to calculate property. And now in my result table, I have a new column here called charge. It will tell me about the charge, what my current pose or ligand has, right? Next up, we have buried molecule surface area. Like I explained earlier, buried molecule surface area measures how much the ligand surface is inaccessible, not accessible by the solvent, right? The better it is, the more it is, the better is for the pose, right? You can just name it. I'll just name it BSA and calculate property. It might take a minute. So if we have any questions as of now, we can address them.
Yes, we have some few thoughtful questions from our audience. So we'll continue and we'll go through them one by one. The first query is, how can I visualize hydrogen bonds and hydrophobic interaction after docking?
Okay, the same thing what I just shown in the demo. For hydrogen bondings, I can just directly right click, look for ligand pocket interactions. And if you want to see the hydrophobic interaction and all such things, you need to consider the surface and what kind of hydrophobic regions are present in around that surface, right? So after that, we have the new column like BSA, which is buried molecule surface area. And by this, we can see how our buried surface area represent in terms of affinity. The more it is, the better is for binding affinity. The next property, what we can calculate from the same panel is volume change. So by volume change, what we mean, we will, sorry, let volume change, name it just VC is enough. So by volume change, what we are doing, we are going to compare the conformations, all the conformation, all the poses here with a low energy conformer. By conforming with low energy conformer, we are generally seeing if there are large changes in that volume, then we can say it is not favorable and it will reduce the affinity. But when we are talking, there is a small change of that volume, we can say it is good for affinity. Any more questions from audience, Selish?
Yes, we have one more query. So the second query is, can I compare multiple docking poses graphically?
Okay, so to compare multiple poses graphically, you can definitely do so. Currently, like I said, we have this volume change column here. You can see the lesser it is, the better for the affinity. Yeah, to answer the question, if I want to compare various poses or a candidate at the same time, I can either use my arrow button or click on array of the column to do them one by one. But for example, if I want to do them do it simultaneously, what I can do, I can use this L column in my result, and let me just hide this from here. And if I use it, I can bring multiple poses in my 3D graphical window at the same time. So I can compare the features. If I want to use a pharmacophore grid around it, I can do the same. You can do it with number of ligands at the same time. You can do such things. So continuing with the more property and description part. Next up, we have plenty of options here, but due to time constraints, we won't be able to cover everything. But there are some important features or properties that I would like to address in this webinar. The next option is to calculate interaction fingerprints. So interaction fingerprints, let me just rename it. It will take a while. Let's just name it IFP. Okay. So by interaction fingerprints, what we mean, it helps to identify the common interactions between all the poses which you just talked, right? And now I have a new column here with which says IFP, and it says some numbers and everything. It is hard to read humanly, but it is very easy when you do it with clustering mode. And to run the cluster mode, you just need to select interaction fingerprint options here, go to cluster, and you need to select this option, the selected column only. When you click on OK, now my table is colored on basis of different clusters, and the same thing we can see in cluster T here. So all these candidates currently in this yellow region showing similar kind of interaction with my receptor. Same goes with this blue, same goes with this brownish pink, and all these candidates individually. Right. If I want to see just one of the candidates from each cluster, what I can do, I can just right click, select centers, and it will select the mid, or let's say center of each cluster here. For example, here we have selected one. So each cluster will have one representative select. And if I want to analyze them again with different tools, I can just right click here, copy or append 13 selected cells to a new table, right? 13 representative from each, each representative from 13 different clusters are now selected, and I can use them in a new table, name them anything, let's say CL1. And these are the representative from each cluster. Okay. The next option regarding the property description will be to look for interaction list. Like while we were talking about interactions and residues contribution and all these things, we understood that the residues around my binding pocket do interact with my small molecule. They have either like apolar interaction, hydrogen bond donor interaction, acceptor interaction. If they have any kind of that kind of interaction, let's just click on IN and calculate property. It will take again a while. And by this, we are saying each residue's contribution towards each pose, and if they are giving any interaction with that, right? Now, previously we have calculated charge, BSA, volume change, interaction fingerprints, and this cluster part. And now, if I see the column headers here, I have interaction of each of the residues. For example, if you see serine 864 is present multiple times here, right? Serine 865 is present multiple times here. Some of the values may give you 0, some of the values may give you, columns may give you 1. So 1 defines the presence of that kind of interaction, 0 means absence of that kind of interaction. You have that regarding to every residue present in your binding site, right? This is again a good option to consider while looking for your good candidates in the screening. And another very interesting feature what we have is hydrogen bond to selection, right? So by literature survey or looking for the looking while the previous let's say in silico work done, we can see there are certain residues by with we can, if we have hydrogen bond interactions, that gives us a better biological activity or better affinity. What we can do when we select hydrogen bond to select option, currently there are labeled residues around my binding pocket, right? So if I want to see any specific interaction with any specific residue, for example, if I want to consider this glycine 863 here, So I want to see how many of my candidates here are making hydrogen bond with this specific residue, right? And that too with this oxygen. For that, I can just select this oxygen. It has given me option. You can just rename it. Let's H bond underscore G 863, the residue ID. Okay, and we can click on calculate property. Now it is calculating the interactions, the poses which are making interaction with this current residue. Here I have that pose. Just wait a second. So it ranges from -2 to 0. Towards -2, it will give me those poses which are making more favorable interactions. And if I go down, you see there are some poses which are giving 0. So there are no interactions. If I can import any of those, for example, this one, it is giving me 0. You see there are no interaction between hydrogen bond interaction between this specific residue and the current pose, right? So this this is just a brief about the post-docking analysis. There are plenty of more tools which would be covered, but with the time constraint, we have not a lot of time to cover everything. For now, if we have any more questions, we can address them now, or else we should conclude.
Yes, Mr. Mishra, we have a last question. How do I export image or a data table from post-docking analysis?
Okay, very good question. I was about to address the same thing. For example, if I want to export my docking results, what I can do, I can just right click to my table header, export it to either Excel or to PDF, or you want to save it in any other format, either SDF, MOL file, ICB binary files, Excel file, and anything, you can do it from here. If you want to export images, you have to do, you have the option to do it with this option where you can make copy the images from your 3D graphical window. If you want to take the 2D interaction diagram, you can directly make that from this tool here, specify the pocket and everything, and it will make a 2D receptor interaction diagram. You can save it to the same, or if you want to present in a Microsoft PowerPoint, if you want to present in paper, anything, you can do it from there. And that label, sorry, this is interactive. If I want to change the position of labels, I can just edit this, shift the labels around. While shifting the labels, it will not change the distance and everything. It will just change the position of the label. Everything will be kept constant, right? And another option is if I want to, let's say, take a video of my current binding pose, right? So what I can do, I can just go in here. You have the video option. You can just name it anything. Let's name it pose 1. You can save in any format. You have multiple options. You can save it, go to this, if you want to move it around, if you want to do any such thing. For example, let me just start the rolling option. And when I click into this record button, it will start recording my screen, right? It will record my cursor if I want to. It will record anything if I want to change the representation, if I want to include or eliminate anything, if I want to make a blob around. Everything what I do now will be saved. This is the smooth transition button, and at last this is save button. And when I go to the same folder, I have the video here, pose 1, right? I can play it, but it might hang the meeting site. Okay. Next up, you remember we made some slides and such things, right? So what is the use of that? If I open my PPT here and switch on developer options by going into this custom ribbon options, downside, switch the developer option on, and at the same time download active ICM from Molsoft website, I have an option to make a interactive slide with ICM. It will ask me which slide or which file I need to consider. So the file, that ICB file which has the slide, you can just take it, open it, and by this it is fully interactive. You can move it around, you can show interactions. It is very good when you are presenting to the audience, to presenting to a group of students and all such things. I also have a lot of slides there. If you have multiple slides, you can just shift between different slides, right? So with this was mostly about post-docking analysis and graphical data retrieval tools, and there are some key things which I would like to add in before concluding. Firstly, common pitfalls that we should avoid is stop blind trusting the top docking pose. So also, so this is the video what I've imported earlier. So this is how it looks when you present. Very interesting. Ignoring protonation or tautomeric state of metal complexes if you have metals in your compound. Overlooking histographic, artifactual, or water positions, ignoring the thermodynamics and all such things is a big no. Accepting highly strained ligand conformations and neglecting off-target, or let's say ADME flags in your ligand or small molecule could result in bad or false positives. So the key takeaways for today's webinar would be to find a good fit. You need to balance electrostatics, water thermodynamics, as well as shape. You need to understand the practical geometry of each pose. You also need to verify some interactions. You can use fingerprints, you can use patterns to understand those. Also, we got to know about false positive, where they come from. We learned about how we can eliminate such false positive, and there are different filters from rapid to advanced filters. Also, we learned something about reporting and documentation using Molsoft tools, how we can get good publication quality images and document videos for presentation. And at last, we got to know about common pitfalls, what we should avoid during our docking or validation processes. So with this, I will conclude today's webinar, and we will have more webinars which are relevant and with more detailed, or let's say with other tools what we have. And to stay updated, you can scan the QR code on screen. It will guide you to our LinkedIn page. We update everything there. So thank you so much for your patience, and I'm glad to present to you. Thank you so much.
Thank you so much, Mr. Mishra, for that detailed and insightful presentation. And thank you all for the excellent questions. Your engagement really enriched this session. So while we try to cover as many queries as possible, some may remain unanswered due to a time constraint. So we understand that, and our team, don't worry for that. Our team will review all the questions and get back to you via mail. So with that, we conclude today's webinar. On behalf of Advent Informatics, I extend a heartfelt thank to Mr. Aditya Mishra for sharing his expertise and guiding us through the practical aspects of post-docking analysis and graphical data retrieval using Molsoft tools. A big thank you to our audience as well for their time, for their active participation and enthusiasm. We are looking forward to welcoming you again in our upcoming webinars. Till that, have a great day and goodbye.
Thank you so much, Arvind.